pride and pleasure in introducing Indrani Biswas. She is uh, MSc from Pondicherry University, where she started from 2015 to 2017, and then from 2017 till this year, where she is currently submitting a thesis, or she has already submitted a thesis in cancer biology, where she is working on very interesting areas of taking, drug designing, cancer biology, phytochemicals, and computer aided drug designing, with vivid areas of interest, largely coalescing, various skills like, gel electrophoresis, forces, RT-PCR, and other things. Her project is titled uh, CADD for anti-tumor leads in lieu of viral diseases, where she is steadfastly, working on methodological developments on host pathological interactions. Prior to joining MSc, she was a BSc honors in molecular biology from the prestigious West Bengal University of Technology. And she has also been a joint secretary over the last 2 years. And she has been a doyen of Bioclues, helping in manifold works in Bioclues, not just, looking into that. So from core point of view, if Indrani is not there, we would miss something. Yeah. So with this little introduction, she will carry forward the meta review part two, that that Manjari has taken forward, and there will be wonderful exercises, and she'll be there for the entire period, taking you today. Yeah. Thank you so much, Indrani, for your time. And today's quick this thing, for those of you who have not joined, at 6:50 or so, after 45 minutes or 50 minutes, Indrani will stop, and we'll be having an online examination. Please give that post evaluation examination in a very fair way, and you will be given about 10 minutes time exactly, and after that the link will be closed. And then at about 7 o'clock we'll start your presentations, where 3 minutes each will be catered, exactly 3 minutes. Indrani will be on time, and you have to stop whatever it might be. And after that, there will be 1 minute Q&A. I'll be muted most of the time, but please excuse me for this. And we'll probably stop by 8:10 or 8:15, and then we can have any feedback orally as well, that you want. Huh? So thank you so much, everyone. Over to you, Indrani. Yeah.
Thank you so much, sir. Thank you for such a lovely and wonderful introduction and that you gave over me. So it's a pride to be a member of Bioclues, and I take this immense pleasure to be a part of a speaker as for this workshop. So as sir has already mentioned, that I'll be going to the part 2 session of the meta-analysis review part, which already Manjari ma'am has covered. So today I'll be taking over you to how to perform this meta-analysis using R. I hope my screen is visible. Can anyone please tell me yes or no if everything is visible or not? Yes,
Yes, it
no.
is.
Okay, thank you so much. So basically, as I've already told you, like you have to take this particular software R. So R has basically two versions. One is the you will find like R, which has multiple versions of it, and the other is the R Studio. Okay, so if you want to perform a systematic review and which you are taking your meta analysis, so if I tell you in brief words, a meta-analysis is basically you perform a statistical analysis of the data that you have collected in your systematic part. Okay, in last class, Manjari ma'am has given you the rather the format how to perform this systematic analysis, right? So once you generate those data in an Excel form, in the form of events and the columns, using this particular software, you have to perform that analysis. So I hope you have all installed this particular software. So, as you search Art Studio, you will see this particular link. Download the Art Studio IDE, and you will get any. Here it is like in 2022, but I'll suggest you can take any versions of Art Studio, provided you have to check the version of the Art Studio that you are installing is compatible with your OS. Say suppose your Windows is suppose 32-bit OS, so the bits you have to take care of it. Okay, and once prior you start with the Art Studio, you have to ensure whether Java is installed or not also. Okay, because these are some tricks which might be faced in some versions of the Art Studio that you want to install or download, or may not be. Okay, so since I have faced these issues, I am putting up these points so that it can be useful for you people also to troubleshoot as and when you perform on your own. Okay, so then so you click on the next, the window will pop up, and then you choose the path where you want to install this particular software, followed by which you click finish and your software will be installed. Okay, so now the thing is, you have your data. You have analyzed your data. You have to put it in terms of some statistical uh, representation. So there will be this kind of plots. I hope many of you are acquainted with these terms: forest plots, violin plots, funnel plots, or
Hello, ma'am.
even hello. Yes,
You're moving your slide or not? It is still uh, because it is not moving here.
it was not moving.
Yeah, yeah.
Okay,
Now it
just
is.
I'll try. You just see now is it happening or not? Just a
Now
minute.
it's in the fourth slide.
Yeah, fourth slide only. I moved it. I was just explaining over you.
Yes, yes. Now the
Okay.
fourth slide is visible. Could you try once moving the slides so that we can confirm the
Yeah,
slides?
just a minute, Dhiraj. Yeah, just see.
Uh, it's not in the presentation mode also.
Okay,
Like once try
just a minute.
resharing again.
Yeah, yeah. Just give me a minute. Okay, just confirm me. I'll put it in slideshow mode. Is it in the slideshow mode now?
One second. Like for me, it is still the Google Meet screen only. Like
Okay.
once again, it's loading. I think. I hope.
Is it fine?
No, ma'am. Could anyone please
No,
confirm?
you have to share this whole screen, Indrani. It's still we are seeing just the
Yeah.
beginning color.
No, I just gave the entire screen, but that is not happening. Okay, just give me a minute. Now,
Yes. Now it is in full screen mode and it is
Yes, now
moving
it's
also.
moving also. It's fine now.
moving. Right. Okay. So, if my initial introduction is it clear to all of you, then I'll proceed with this the upcoming slides. Then, shall I proceed?
I think you can once show that installation wala
Installation part.
one also.
हाँ
Yeah,
ठीक है ठीक है.
you can
Okay.
show it
Yeah.
right.
Sure. Yeah, yeah, sure. Okay. So the thing is, yeah, as I was mentioning you, so the R Studio. So as you type on Google, so you will get this particular several hits. Download the R Studio
It's not.
IDE.
Acha, okay, okay. You can show. Thank you.
uh, Download R Studio IDE, and then you can just check below the different types of versions that is there. Okay, so whatever version you have got, you can just you will see free download one option will come. Okay, so below that one free option, so you can just click on that, and the download as it starts, one pop-up window will come up where you want to install it, like that is for the directory that is asking. Okay, so once you do that, you can click on finish. Your software installation will be over. Now one of the thing that you might encounter is after installation is based on the OS that you are having. Is it 64 or 32 bits? So based on that, it's better if you have Java has been installed or not. Okay, so once you get this systematic analysis, like you have accumulated all the data in the Excel format, and you have finished all the analysis part, now you have to represent the data in terms of your significance, like which study has been more significantly reporting the topics that you have chosen for, right? So to represent this, you have several types of graphs. Okay, so those are like forest plot, funnel plot, volcano, and violin. Okay, so if we take a forest plot, you will once you have run the analysis, you will get something like this box kind of thing, and in between there will be a line, and you can see for each type of these studies, this width will be changing. Okay, so that represents actually the confidence interval. Okay, so I hope you are all aware of the terms confidence interval. So that is basically the 95% chance that your studies will fall in the range of negative to positive in both the side both sides, two-sided test, because you're choosing a p value, right? So it will have the probability like 90% or 95% chance that your studies are falling within this range. Okay, so as you can see, each symbols I have just put it up here. So what does it indicate? So basically the midpoint of the box is actually your mean effect of the study. Like you take an average, so many studies you get performing a systematic analysis, so you have to take a mean estimate of that. Then followed by the area of the box, so which gives an weightage for a particular study, and diamond will represent an overall effect estimate. So if once you run see, when you do the practical session, you will be getting a clear idea how this particular plots are helpful for your study point of view. It depends on the type of the analysis that you are doing and the type of the plots that you have to represent. Okay, so the other plot that you can also represent your data is a volcano. So it's looking like a V-shaped kind of thing, or like a volcano means all your data sets are being haphazardly distributed. Right, now here one very crucial thing is this kind of data set, mostly you want to check the statistical significance of, you want to say suppose 100 of genes, differentially expressed genes you are studying for a particular disease. So how do you expect that, how you want to correlate your study with in terms of statistical significance? So you have, in gene expression studies, you have something called fold change, right? So that represents like to what extent your genes are getting upregulated or downregulated. But even though if it is getting upregulated or downregulated, but how do you classify it is you have to say it is significant, you have to estimate this statistical significance in terms of p value, right? So here you plot in terms of the fold change, that is on the x-axis, and you have an y-axis, that is the negative logarithm of your p-value. So whatever points which are lying above this red line, basically it will be giving your statistically significant value. Okay, so here I've just given one example, like for genome-wide association studies. Say, suppose you are trying to understand a distribution of a particular gene in terms of populations based study. So there you cannot apply any thing called like parametric test. You have to go for the non-parametric, right? You cannot assess a mean, median, mode. So you have to perform this chi-square, and you have so many samples. You have say suppose beyond 300 400 samples. So how are you going to analyze the population frequency of that particular gene? So you have to take plot this kind of plots called as volcano plots. Okay, so next, I hope you have came across this term in the previous presentation with Manjari ma'am. So it's a violin plot. Basically, it is like a modified version of your box plot. Okay, so a box plot, you know, right? In the middle, we have something called median, and the upper and the lower interquartile range. So this kind of plots again, you can plot based on the samples size that you get. It's, you can say like we can also put box plots, but that is not applicable in case where you have numerous samples and you want to study the median distribution of your samples. Okay, so it will be giving you a summarized data, a summarized a value that can represent your basically that you the data that you are going to interpret. So this is the other thing that we have called a funnel plot, because you can see here it's in a typical funnel shape. So values which are lying in and across both the sides, you will be estimating the measurement of the study or the size that is there in with respect to some odds ratio and the standard error. Okay, so odds ratio is again applicable mostly in case of the clinical study designs that you take. Okay, so these are the three like few things that I want to mention, that before you want to take any systematic analysis, meta analysis you want to perform on your systematic studies. And few checkpoints, yes, as I told you, you have to confirm the version of the R and the Java. And packages, I will if I'll tell you while we do the practical session. And the plots that you are going to design based on the clinical samples or the type of the intervention, like the experimental design of your study, or the types of the methodology that you have put in your studies. So that varies as the type of the plots that you want to represent for your particular systematic analysis. And yes, this is a very crucial criteria. You have to put your events and the columns, because that is how if you want to interpret your meta analysis data in R, it will be able to read this. Okay, and so if you do this particular meta analysis, it is basically a qualitative as well as a quantitative analysis also at a same point of time that you can perform. Okay, so now that I'm done with the presentation, I just want someone to who have installed the software and can share your screen. Anyone? Pranita ma'am or Jeshi, if she is here, I don't know. That will be easy because to follow in everything in step by step manner. Okay, Jeshi has not joined. Pranita ma'am or someone else?
Mm, yes, I'm having installed
Yeah,
R,
sir.
but
Yeah, yeah.
it is long back. I think it is installed, so I don't know means it is having your package or not. But
We can do that, sir. You have installed R Studio, right?
yeah, R Studio.
Yeah, yeah. Please share your screen, sir.
Okay.
Okay. So, sir, you have also have that Excel file, right? that I've given
Yeah,
it to
I'm
you?
downloading. Yes.
Okay. Just before starting, I want to tell something like you create a folder, say suppose in your desktop, and paste that Excel file in that particular folder, sir.
Okay.
Yeah. So just make sure you don't have any other files in that particular folder, sir.
Yeah, I've just
Yeah.
prepared new one.
Okay. Okay, sir. So can you just open that Word file that I've shared you in
Yeah, yeah.
today?
Vipul, you have Java in your system, right?
Yes, it is there. But
Yeah.
I think
Okay.
2 3 months back I used it just for training
That
purpose.
is
No,
fine.
if it
That is fine, sir.
installed is that's more than enough. Yeah.
That
Yes,
is
yes,
enough, sir.
yes.
Okay. So, see, sir. So the first thing is what you have to do is you can go back to the art studio, sir.
Yeah.
Yeah. So firstly is we have to open the directory. So you can go to the session in the top taskbar.
Hmm.
You see the session. Yeah.
This one.
Yeah. Click on that. Next, you see set working directory. Set working directory. From there, below it is there. Choose directory. So that particular file, that Excel file, you have kept which
Hmm.
place,
Yeah, it is on desktop
desktop,
only.
and
T.
just open. Yeah. So just click open. So this is the first step. That is, you have to choose the directory where you are storing your files, and make sure no other files are present in your that particular directory. Okay.
Okay.
Okay. So before we start, see below this we have number of packages, sir. You can see, right?
Yeah.
Yeah. This blue, yeah. Those list of the packages. So packages are basically sets of some. It will have some commands and some sort of functions that will enable us to perform this meta-analysis. Okay. So, sir, I want you to install Dev Tools. DEV.
DEV.
DEV. You can just scroll down or you can just write there.
Give to this one.
Yeah, so just click on that and click on install.
Um,
Just checkbox. That's it. That is enough. Checkbox that in the
uh,
packages you have looked into help packages there only.
packages.
Packages, yeah.
This one. Okay.
Yeah.
Yeah,
Yes,
just click.
that's it.
Uh,
Yes, it's done. Next.
Okay, next we can go for metaphor and meta, sir. You just type metaphor,
Meta.
META. Yeah, I can just type in the chat box, sir. Just a minute.
Okay.
Metaphor.
Search box. Just now where you search for DEV.
Yeah, okay. Okay. Here. Okay.
Yeah.
Meta.
Okay, you haven't downloaded the package, so you have to install the package.
Okay. So in install, I have to.
Not here. Yeah, from here also you can do that. Try
Yeah,
Uh,
you can do it, sir.
install.
You can just you can
Yes.
click
Yeah.
metaphor first. METAFOR.
Okay, change that format. Install from instead of package,
CRAN.
repository. Yeah. Yes,
Yeah, cran. Yeah,
Uh, Meta.
that's it. That's the
FO.
first one.
Okay. Meta 4, no?
Meta.
F4.
Yeah. This one
This one.
followed by the meta. You can go both.
Both means
Install.
first.
Yeah,
First
install.
install this.
First. Okay.
Let it run. Let it run. After that, you can search for another one.
After you install the packages, just wait for some time. It will get automatically updated. See, those commands are the red color lines that is indicating those packages are getting installed in your this version.
Excuse me, ma'am. What was the first package we installed?
Dev tools, sir. Just a minute, I'll type in chat box,
Yes,
sir.
sure, ma'am.
Now next is Meta for Forest.
And wait, it's still
Oh,
installing. You shouldn't
It's
do that. It will
installing.
overwrite.
oh,
You just cancel this because there will be sometimes, certain complete upgrading of other packages will happen. So wait until you install this. Yeah, cancel that. Click on cancel here.
yeah.
Yeah, that red symbol should stop.
Okay. Now
Yes, it's done. Now the next one,
Yeah,
Meta,
you can Meta, only Meta.
only Meta.
Yes, sir.
install. Yeah.
So before you start with the command sets, you have to make sure like all these packages should be there, or else when you
Yes,
run one
click on
after the
yes.
other code, you will get errors in between. So be careful with this packages, and it depends on the type of the analysis. This is for the meta analysis. You will get to know a lot of packages that is there. So based on that requirement, you have to check which kind of tools that you or the packages that you want to install. So it's loading actually. It will take a bit time.
Is there anyone who doesn't use R here?
Is it over or is it still running?
Yeah,
It's
it
still
is.
It's
running.
still it's running. It's running. Okay. Okay. Yeah.
Only those three packages, right? Meta.
Yes, Dheeraj. These three
Yes, ma'am.
packages, after all these packages ends, there will be one more package that is very essential if you want to perform this meta analysis, because that will have all the commands that you want to develop the each type of this plots that we want to develop, right? The forest plot or the funnel plot.
Yes, yes.
So there will be one more package after the installing ends. We can install that one. So then only you can start with the commands,
Yes, ma'am. It is
or else you
completed and
won't, or
binding.
else yeah, or else you won't get the complete the plots that you want to, or the graphs that you want to plot against your systematic analysis.
Now it is completed in this as well.
Yeah, I think it is done.
Yeah, so it is coming download. Okay, so now the next one will be qdap.
Q.
qdap qdap
Install,
tools.
install. I don't think you will have it.
Install.
It install.
You have to install it. So I've written in the chat box QDAP tools.
No,
DAP.
it's just QDAP. That's it. That's it.
QDAP.
Yeah.
It's just QDAP, right?
Yeah.
Ma'am, actually for uh, both the things, I like for my package it was like both the
Okay.
things were required. Yeah, it's
Okay.
better to install both of them.
Yeah, I think it is done.
Okay, so next you can go with the QDAP. That this will be the final package that you will be installing.
So QDAP means which we have installed just now. Huh?
No, that is the tool,
Again,
sir.
again install. Again install QDAP.
Again
You
install.
had earlier.
Again install.
Okay, this one.
No, no. You have
Okay,
opened the library. There we
okay,
need something called QDAP also.
You just type QDAP, it will come.
Yeah, that's it.
See,
The
see the
first
first
one.
one. The
okay.
first one.
QDAP. Okay.
Yes,
This.
yes. So install. So all these 4 to 5 packages, it might be like in some versions of the R Studio you may not require, but it's better. You can install all of these packages and you can start your commands. So I've listed the order of the packages that you have to install. In that order, you can follow it. This will take a bit time because it is the most
Oh,
like a bit bit, it's quite heavier as compared to the other
Lengthiest.
mm-hmm.
compared
Lengthiest
to
to
the other
install. Yeah.
packages actually.
Okay.
It has the complete setup that you require to prepare one meta analysis. That's why it takes maximum time.
It's done.
Yeah.
Now click on all those ones which have downloaded.
Okay. One is QDAP.
Yes, QDAP. No. QDAP should be the last one.
Okay. Meta.
The first one would be
Dev
Meta.
tools,
Yes. DevTools. It's done.
dev tools,
Check.
Meta.
Hmm.
Meta. Meta.
Metaphor. Yeah, you
metaphor.
can do that. That's fine.
All three.
Yeah.
Yeah, it's okay.
Okay. Then one
Okay.
more.
So
Means
yeah, the last would be the Qt app that you install.
QDAP.
Yeah.
QDAP tool and then QDAP dictionaries. This one.
No, sir. The Qt app tools is there, right?
Yeah,
I
tool is
think. Yeah. Okay.
last
So
one.
I
That
think
one is I think QDAP only.
tools. Okay. Okay. Fine. Fine. This should
Okay.
be fine. Okay. Let's see. So, sir, can you just open up the Word file, sir?
Okay.
Yeah. So you have to now import that Excel sheet, sir. So you have selected your directory. You have to import the Excel sheet. So you can go back to the R Studio.
Hmm.
Now in top, there will be an option to for importing.
This one. Oh, this is open. Import dataset
Import data set. Yes, yes, sir.
from
So you
Excel.
have to. So it's an Excel file. Yeah. So that Excel file where you have restored, just open that Excel file. Browse. You can go and browse. Yeah. Just which folder you have saved. Yeah. Click on import.
Yeah, it is import now.
Yes. Yes. So as it is imported, you can see that that particular file is getting viewed there. Okay, so so go back to that Word document and type the first command that we have.
This one,
So
no?
files, yes, you have to type. It's better to go for typing. You can,
Okay.
because in paste copy pasting sometimes we have some errors, sir.
Okay. So this yeah. Means again I have to I think open side by side.
Okay, yeah, you can do that. That will be easier for you to type
Hmm.
the commands in, sir. And make sure each and every comma space or the enter that you're giving, you should be very careful with that. uh, with that also you'll be getting the errors. Okay.
Sorry.
Yeah, so you can do control Z, sir, control Z.
Uh, yeah.
Yeah, so you can just minimize it and you can do it.
Oh, sorry. For side by side, it is sometimes I this hist dot files. I think it is space, no?
Pattern space equals to,
Is equal to
then again space.
okay.
So
Dot
inverted
XLS
comma dot xls x.
x and this comma over. Okay.
Inverted comma closed comma, then full dot names
Then comma.
equals, yeah.
Oh, sorry. Okay, or it is, I think this
Okay,
one.
so just
Yeah,
cross check it, sir.
yeah, files, file list of files, path, space, xls, full dot name, full dot names is equal to t comma cursive. I think it is okay.
Yeah, please check the space, the comma, and the
Hmm.
everywhere that are included, sir. Yeah, so go on to the next command, give enter, and you can go on to the next command, that is
Try copy pasting that one
Yeah, yeah,
just
I
from
think that
files from the Word doc and just see that. Maybe it might work.
otherwise.
डबल वो ग्रेटर देन सिंबल 2 बार आया, एक ही बार होना है.
Okay, this one, na? हाँ,
Yeah, yeah,
yeah,
yes, done successfully.
yeah, yeah. Now,
Next, go with the next one. I
next,
is equal to 1.
yeah,
Yes, yes, right.
I is equal to 1.
हाँ, एक के बाद एक कीजिए, एक के बाद एक कीजिए. ये CS
हाँ भाई,
CS नहीं नहीं CS पहले कीजिए, उसके बाद I is equal to 1 कीजिए.
नहीं, that is already done.
Oh, it's done. Okay, okay, fine. I've been zooming it from my phone, that's why.
Actually, by side, actually means I
हाँ,
have to
और
also.
यहाँ पे वो प्लस सिंबल नहीं आना चाहिए था. फॉर द नेक्स्ट वन दैट प्लस सिंबल यहीं पे निकाल लीजिए.
Okay,
इन द वर्ड
like this
इटसेल्फ
one,
रिमूव दैट. हाँ, यस, backspace, that's it. Now copy it.
hmm,
Hmm.
is okay.
Yes. Okay, there's का error आ रहा है.
This same actually here also. Same thing is
Yeah,
there
okay.
in case of
Yeah.
so maybe one more time you can try installing the Qt app, sir.
QDAP. Okay.
Qt app, yes. I don't think it got installed
और
properly.
इंद्रानी जस्ट अ सेकेंड, आई
हाँ,
पोस्ट वन
हाँ.
डायरेक्टली. वो डायरेक्टly भी यूज कर सकते हैं. लेट्स ट्राई द डायरेक्ट वन,
Let's
अदरवाइज
try
देन
with
वी
that.
कैन डू
Okay,
दिस.
yeah, sure, sure,
Yeah.
sure. Sir, just cancel it. Let ma'am put that link, command.
Yeah.
We can try with that. If that doesn't work, we can go with this alternative.
I think there is a plus here, which we have removed. Huh?
No, actually, that command, so usually it comes when the Qt app is not installed properly.
Okay.
We'll try
नो
with
नो
the
एक्चुअली
alternative.
नहीं नहीं, डायरेक्टली क्या कीजिए, यूज द मेटा वाला, द वन आई सेंट इन द चैट बॉक्स. यूज दैट कमांड डायरेक्टली, द मेटा वंस इन द चैट बॉक्स.
This
Yes,
one up to meta. Na?
yes, yes, yes. Up to that data, up to the data. Data ऊपर, हाँ हाँ, yes. I hope it's the same file name, right? The Excel you had taken.
Yes, yes, yes.
Yeah,
It's the same
just
file that I've given you.
copy paste it and just enter. Yes, it's done.
Yes, it's done.
So the next one also, yeah, next one also use the next command which I pasted in the
Okay, that would be better if you typing from means that side.
yes. Now that PDF wala,
Yeah, yeah, this one. Up to
yes,
this one. This
yes, 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 yes,
250.
yes. You have to do it stepwise. Enter. Okay,
So
DR
in
is
that,
not.
no, instead of slash, sir, you should enter the file name data that I have given you. The Excel file name that is there.
Yeah.
Instead of the slash, you should enter that
Yeah.
Okay. Okay.
and remove the next dr. Then it will come.
This one, na?
हाँ, वो जो slash है ना, sir, उसके instead of that, you have to put the file name.
File name
You
here.
paste. Yeah, they
Okay.
will paste the same command and then change the command.
Okay. Okay. File name means this one, na? funnel.
No, not that one. File name is used data underscore brew2.
Okay. Here, no?
Yes, sir. Yes.
Means can you speak or I have to
Da-
search?
I'll just type in the chat
DATA
box.
data
Uh,
Capital
data
underscore
D,
and
capital D, capital D.
capital D. Okay.
I've
Data.
written in the chat box, sir. You can just copy paste
Okay.
that.
Underscore
Okay.
yeah,
BI.
yeah.
That's good. Okay. Now
Remove the
Yeah,
dr.
just
The
remove the dr, sir.
this one or earlier one. This one or this one.
Yes, that dr, sir. That dr. Yeah.
This one. Only.
Yes.
Okay. I have
Comma.
means again
Just a minute, sir.
comma भी निकाल लीजिए.
comma भी निकाल दूँ.
हाँ,
Is okay.
yeah. Now
Yes.
enter it. Now
Yes.
if it's working, it should come. The other dr also maybe you have to remove.
Okay.
You can use the symbol, up symbol, up arrow. It will automatically copy your
Yeah.
previous. Yeah.
Hmm. This DR
Yes.
Yes.
now.
Yes. Now enter
Okay.
it.
Hope this works.
Yeah.
Yes,
Yeah.
Yes.
it
Yeah.
worked. Done.
Good.
Done.
Done. So the
The
next
next.
command.
Next command. I think this one. No? For
Forest
Yeah,
meta.
forest
Yes.
meta.
hmm,
Yeah.
this one. Null
Yes.
to one.
No, only till
Dev
off.
dev dot off. Only that command, sir. You copied.
Okay. This one only.
Huh.
Yes.
Yes. Yes.
Okay.
The
Null.
So
last
you
one.
can just open your, now that this is done, your forest plot will
और
be
एक है
generated
और एक है
automatically in that.
funnel meta
Yeah,
वो भी कर
we'll do
लीजिए.
with that
Done.
also. Yes.
Okay. Yeah.
Yes. Now you
Let's
can
see.
look into that
Yeah.
folder that you had
Just go
earlier
to the
saved your
folder, sir.
files.
Okay. This one. Yeah.
Scroll down, sir.
Yeah.
So you can see your forest plot is generated, sir.
Okay.
So against each type of studies, how many studies have been reported on the type of keywords that you mentioned for your search, right, using those databases, and your confidence interval, like to what extent that particular study is showing your the efficacy of those terms that you are searching against your topic. So this way you can interpret, and you have got the other one also, that is the funnel plot. So
So
however you want, you can now just interpret this study, sir. Now,
okay. Now I have to go to again R Studio or shall I?
These are the files you usually see in, or the plot and this image you usually see in papers also. So these you can use and then write the results and discussion based on this.
And madam, for this raw data which we have submitted, means in earlier
Mm-hmm.
from Excel file,
Hmm.
in that particular means file, just we have to give a reference only. I mean,
Yes, this is the format in which you have to give the file in the for R to run the meta
okay,
analysis.
okay,
Okay, the first study, say suppose you had 26 studies earlier, Pranita madam had, like, so first study. Now, what does it say, that what are the total number of subjects and what are the events which had the positiveness which you are looking for? So you
okay.
keep on adding all these and then generate the forest plot to see a cumulative effect. सबको मिलाने पर क्या होगा? I mean, like, what is the effect it's coming? Is it the same? Is it different? That is what you'll get in that forest plots.
basically, all three plots are like scatter only, sir. If you see, it's like a basically scatter plot. The distribution of the data will be different, the way you want to represent it. That is a fundamental concept, sir.
Okay.
I hope everyone is clear with it, and we have given the commands, so you can just try with that, and you can reach out to us if you face any further errors also. Right, ma'am? Is that will that work, right?
Yes. I think we have to give time for now for these people to
Yeah,
write their exams.
so write their exams. So we can, I'll just drop the link for the exam that Prash sir has mentioned in the WhatsApp. So give me a minute.
Yeah, I think you're done, right, Indrani? Yeah.
Yes, sir. Perfectly, sir.
Yeah.
We
Thank
are
you.
done
Thank
with
you so
this,
much,
sir.
Indrani, for a wonderful talk. I think Indrani deserves a wonderful round of applause. Please give a
No,
big round
sir.
of applause, and not the
Manjari ma'am also has supported me a lot.
applause,
So I
but
should I should reveal one secret. So I've just learned this
great
programming within
applause.
just
Yeah.
4 days. Yeah. Thank you, one and all. Thank you, Precious sir. Over to you, sir. Vipul sir, you
Vipul, you
can
can stop
stop
presenting then.
presenting. Yeah. Mm.
Okay, ma'am. Yeah.
Right. Um,
So the in the Word document also the whatever file codes were given, it should also work properly. However, what I did was I tried to remove the troubleshooting and make it as simple as possible. So we'll share whatever code we have given in the chat box also in the WhatsApp group also. You can later try that. So right now, please go on with the next session, please. Okay. Yeah. Thank you.
Prash sir, shall I share the link in the WhatsApp group?
yeah. Uh, th- yeah. So thank you, Indran and Manjari. So all of you, please switch on your camera, for the examination, please. This is a must. You all have to switch on your camera. Yeah.
I've shared the link in the group.
Yeah. First, let not in the WhatsApp here, not in the WhatsApp, in this group, in this chat,
Just a minute, sir. Yeah.
not in the WhatsApp, Indrani. Indrani,
Prash, I'll be leaving. I have to go out. Okay.
Indrani.
Yes, sir. Yes, sir. Just a minute.
Prash, Prash.
Yeah, I have shared the link in the chat box. Kindly, all of you can switch on your cameras. Suresh and other people also. Suresh, can you just kindly switch on your camera, please?
Yeah. So Indrani, please do not share the link in the WhatsApp group. It's only here in the chat. Yeah.
Yeah, yeah, I've dropped it here only, sir.
Yeah. So all of you, please switch on your camera, and you have about 8 minutes, not 10 minutes, for this. So Indrani, now you can, if everyone has switched on the camera, please consider them to be
No,
qualified for the examination. Yeah. Please share the link,
yeah,
and you can count in exactly 8 minutes. Okay. Yeah.
Sir,
yes, sir.
I'm on the
Timer.
way to my lab. Please
Yeah.
please give me
No.
2 more minutes extra.
Sure. Sure. Yeah. Please share the link, Indrani. Yeah. Thank you. So you have the link that is activated until 7:05 p.m. Please. Huh? Thank you. You're Indrani, huh? Dance, right?
చెప్పు మీకు తనకి వచ్చేసామని వచ్చి మెసేజ్ ఇవ్వచ్చు కదా డౌన్
Look, 5 minutes to tell them. Exam. Yes, Suresh, Pooja, Malvika, please switch on your camera. Suresh, the other Suresh, please. Yeah. Yeah, Ankita and Pranita, please switch on your camera.
Pranita ma'am, uh, please kindly switch on camera.
Can someone share me the link?
Just one more minute to go.
Yeah, the time is up,
Okay,
please.
time
Yeah.
time is up.
I don't know how much I used it.
The link will be automatically closed in 1 minute, please. Almost it's done. Please, you have to submit it. Otherwise, you'll not be able to submit the answers. Okay, the link is closed. Now, please start the presentations. Indrani, please give them 3 minutes
Sure, sir.
each. Okay. Yeah. Thank you.
Yes, and okay, sir. Fine. So, who will be first to present? Dheeraj, are you presenting? Firstly.
No, I
I
think it
asked
is
for
Yasin. Yeah.
Okay, okay, ma'am. Please, please, please. So 3 minutes for presentation and 1 minute for QA session.
just a minute. I'm looking for it.
Yeah.
I'm just
Okay, okay. Please.
Can you see my screen?
Yes, ma'am. It will.
Yeah, ma'am, you can just yeah, please
Uh,
start.
they are all very good afternoon. Uh, I was trying to identify some genes in a genome-wide mode, but the thing is, we have not yet completed the complete identification. So I have just given the gap. Means it is yet to be completed. So once the inference is derived, then I will write the name of the genes. So this is the idea. Um, my slide is not moving. Okay, yeah, it's moving. The background is already we have done some work in Hasgram. We have taken some stress responsive contrast accessions identified from total gene bank collections through multi-location evaluations. Using this sample, we have gone for transcriptomic study, and then we have identified some contrast, and then LNC also we have identified, and then microRNAs also we have identified. Then we have identified the targets, actually which microRNA is targeting which kind of gene. So from that, now I have picked only one gene for the present analysis, and I am trying to identify the linked genes or co-expressed genes, which are all also regulated by the same group of NIRS. We have identified total 15 microRNAs, which are actually regulating the stress response in host gram. I want to find out the same thing in the co-expressed genes. So in this method, actually the transcriptomic work is already done. Earlier we have completed that. So now I am trying to pick only one gene. That gene is ethylene responsive factor. Okay. So that factor, that nucleotide sequences I have taken from soybean, and then I have studied the domain. I have checked the domain, and then I identified some network analysis. Means I have done some co-expression. Means I tried to find out some co-expressed genes for that. Then for the same protein, I have developed some model and then validated that model also. So up to this, yeah, I have done. Leftover is the non-coding RNA prediction and the other targets I have to find out. Meanwhile, I have taken the phenotypic data and then I grouped that into 4 classes. Actually, I have developed one core collection from the total gene bank collection. So I just want to validate that with this Vega tool. And then I have plotted the total variation like this. So here you can see the 4 classes spotted like this, and this is the diagonal one, that is like a correlation, and this is one group. Actually this group is very distinct, so in every plot as almost all plots we can find out that as a distinct one here. So that is the inference. So the conclusion is work is not yet complete. um, so after analyzing the co-expression network, and then again I do the LNC and microRNA kind of work, then I will be able to find out what gene I can fill in the data. That's all. Thank you so much.
Okay, thank you, ma'am. It's quite extensive work. So, anyone is having questions? Raj sir? Anyone else from the participants? Okay, I just have one question, ma'am. So, the transcriptomic analysis, what is the software or the tools that you're using for the transcriptome analysis that you did?
Actually, I started actually that transcriptome I did earlier before this work. The top hat and other boutique kind of thing. The same kind of order we have used. So almost the same we have discussed it here. So, but the analysis I did with the de novo, and then I compared that with the reference-based assembly also with the soybean. So that work I did earlier.
Okay, thank you, ma'am. Thank you so much. So, yeah, I would request the next speaker to share his or her presentation. And Dheeraj,
Yes,
who is next?
Monica, I can
Who is
go ahead. Okay,
Rajeshri? Wait, yeah, has raised her hand. Yes, sir, please. Any questions? Rajeshri, can unmute yourself and speak out?
I'm audible, ma'am.
Yeah.
Yeah, please.
Okay, so can I present?
Oh, I thought you are asking question.
Yeah, Rajesh,
No,
Dheeraj, shall we proceed with him and then
Yes, no
you
issues,
can?
no issues.
Sure,
We
sure.
can.
Please kindly share your presentation, Rajesh.
okay, ma'am. Is it visible, ma'am?
Are you presenting from a mobile?
Yes, ma'am.
Okay, please proceed. So you have just 3 minutes time, sharply
Uh,
9. Yeah, please start.
Okay, so I have done an in silico SNP finding, a whole exome study to find how the translocation occurs in chronic myeloid leukemia. So basically, chronic myeloid leukemia is a blood cancer which is happens due to translocation between BCR and ABL genes, and we can see the working in the next slide. So this is the BCR and ABL. So BCR is found in chromosome 22 and ABL in chromosome 9. So what
Sir,
happens is
sorry to
during
interrupt. Like,
translocation, the
one
BCR
second.
breaks up and the join.
Just a minute, Rajeshri. Yes, Dheeraj, please.
Sir, could
Yeah.
you please turn your landscape into landscape mode? We can uh, visualize that also. Please, if you can
Uh,
do that. You can rotate your
yes.
phone. It would be fine.
Uh,
I, I don't think he will be able to present,
okay.
Dheeraj. If you just find if any rotation option is there, just see if it
Yes,
is
yes,
there or
yes. It is
yeah.
fine.
Please, please. Great, great. Please carry on.
Okay. So this is BCR and ABL gene fusion. So translocation BCR and ABL gets fused, and that's why CML gets occurred in individual. So the basic question is, we all have BCR and ABL, and we know that their fusion is the only reason why CML is happening. So how does this translocation gets triggered? That's what I'm finding. So that's the objective of my project. So the this is the basic methodology of whole exome sequence pipeline, where we are taking data and doing the necessary steps. I hope it's visible. Mm. So what I found is, using the whole exome sequence, I found many variants which are essential for this translocation, and from there I got some top genes like PLK3, AT, and JAK2. So these are the genes, and on the right side you can see the network from Stream database. So this is the hub gene result from CytoHub, which I did it in Cytoscape. So it shows the JAK2, ABL1, and 18. These are the highly clustered genes. So the final results is, the top genes are which are potential putative drivers are PLK3, KM2TC, and FGRD, this one, and which are already reported as JAK2 and ATM. They are already published in many papers related to CML. And the other two genes which are very less research is PAPBC1 and GXYLT1. So in conclusion, I found some genes which I already told that which can be mutable mutation driver. So I still have to validate them. It is not a complete paper, and which are already published, like the ATM genes. So I can say that my work is kindly in the right direction, and I hope that I will have to do many future works in experimentally, not only theoretically, so to validate my results. So thank you. This is all. Any questions?
Yeah, it's very nice presentation actually. You have very good idea of preparing a presentation, but make sure next time onwards, Rajeshri, just try to present it from a laptop, okay? When people are watching some
My
presentation, they
laptop
expect.
is not working now.
Oh, okay, fine.
That's
Just
why like I
it's okay.
have to do it in mobile.
Oh, it's okay. Fine. So, anyone have to ask any questions from participants? Yeah, well, only 5 participants have submitted the exam. Strange. Okay, so we'll proceed with the next presentation. Rajeshri, can you kindly stop sharing your screen?
Yes, ma'am, I stopped it.
Sure, Prash, sir. Anyways, it's been
Yeah.
recorded, so we know. Harsh Chowdhury wants to say something.
May I present?
Okay. Sure, sure, sure. Please.
Yeah, just 1 minute. Let me share my screen.
Dheeraj, maybe let all the participants present, after following which you can present, Dheeraj.
Yeah, sure, no issues.
Is it visible to all of you?
Yes, sir. You can make it in
Yes, sir.
slideshow mode, sir, please.
Yeah, yeah, yes. Is it better now? Should I start? Am I audible?
It's
Am I
pretty
audible?
good. Fine.
Hello.
Please,
Am I
please.
audible?
Yes, please.
Okay, thank you. A very good evening to all of you. My name is Harsh. I have completed my bachelor's in biotechnology honors from IT University Gwalior. So I have done a small study that I was very keen to do. It was like investigating the role of ERBB2, a protein in the cancer progression using integrated system bioinformatics. So if I talk about what I have done, is it moving? Yeah, okay. So here the things that I have done. The first thing that I have done is I have done the protein-protein interaction analysis using Cytoscape and Cytohava. Then I have downloaded the SRA of whole exome sequence of colon cancer patient, one from for a male patient, other one for a female patient, and I have done the fast QC analysis using Galaxy tools, and I have found something that I'll be telling you. Then I have downloaded a lung cancer data set from a Kaggle website and I have analyzed it using Veka software. Finally, I have proceeded for the molecular docking of ERBV2 with Windolene. That is a part of my major project research work in university. Finally, I have analyzed the results of molecular docking using Discovery Studio and Camera X. And I have also came to a conclusion that future work is required that we'll discuss. And when we talk about this ERBB2, actually ERBB2 is a erythroblastic oncogene, which is a part of a human epidermal growth receptor family. And overexpression of this ERBB2 can lead to various kind of cancers. You know, some of the most common types like breast cancer, ovarian, colon cancer, lung cancer, even gastric cancer is also there. Okay, and this ERBB2 is also known as HER2 or new. It is encoded by a gene present in the human genome on chromosome number 17, I think Q21 or Q20 position, that is ERBB2. You can see the PDB-ID 2A91, that is the crystal structure of ERBB2. Now let us talk about the resultant discussion. So here we can see a network map of the PPI interaction of ERBB2 gene. Okay, and all of the genes with which this ERBB2 protein or this ERBB2 protein is interacting, all these, somewhere they are related to metabolic pathways, which if not going to work properly, they will lead to a kind of a cancer. Okay, so all these genes related in this map are responsible for a kind of a cancer. So this was the analysis for Cytoscape and Cytohava. Then if we talk about the WES, whole exome sequence analysis, I have used two sequence, male forward sequences from male and female. And I come to know that the male patients suffering from colon cancer, they have higher GC content than the female colon cancer patient. I'm still finding out the reason why. Okay. Then if I talk about Veka analysis, so when I analyze the result, I came to know that people, who are more prone to alcohol and they are more smoking, they are more prone to the lung cancer. And in them also, as per some studies published in 2011 and 2013, ERBB2 is somewhere responsible. Finally, I have done the molecular docking. Here we can see some protein interaction and some of the amino acids. Then we can see 2D diagram. We can find, some conventional hydrogen bonds are there. Okay, and carbon hydrogen bonds are also there. I have done more analysis, but due to restriction of time, I'm not presenting. Then, if I want to conclude, we can say that ERBB2 signaling pathways are one of the most important pathways that we all need to study because it may lead to various kind of cures that may be for various kind of cancers. But all the the research work that is going on now is focused on the target and identifying different kind of therapies. But we are not focusing on what is the effect of those therapies on other metabolic pathways going on in our body. because when we talk about human body, it's a kind of a system. Everything is interacting. So after identifying the research work, I have came to the topic that I would love to work in the future. That is like genome-wide analysis of ERBB2 signaling in cancer, insights in the role of ERBB2 in different cancer subtypes, and the potential off-target effects of ERBB2 targeted therapies. Thank you. That's all from my side. If anyone is having any question,
Yeah, very nice planned study, sir. I would suggest. So, if you look into this as a future perspective, as I mentioned, it could be very useful, I hope. And I've seen all of them, all of the participants who were representing have utilized all the tools and techniques that were taught during the training session. Very nice to see that you have all learned and enjoyed this training period. Okay, so any more questions from anyone else, or else we can continue with the next presentation. Yeah. So Ziaul, is that you going to present now? You can share your screen if you are here. Ziaul. Okay. Next, if he's not responding, maybe it's a network issues. Next, who is it? Okay, Ziaul. Fine. Vipul sir, you want to present?
Okay, if he is there, then okay, no problem.
No, he said he will present later, so you can continue, sir.
My slide is visible.
Yes. Yes, please, sir.
Okay.
Please put in slideshow mode and you can continue presenting.
So this is the presentation I made out of one of my means intended study. That is on molecular investigation of plant architecture in PGNP, that is ARHAR, through omics-based approaches. Basically, in PGNP, plant architecture play a very, very important role, because plant architecture in terms of dwarf and tall, this is not the way, I mean, this is not on the two, but there is a compactness and there is openness. It is also makes difference in case of photosynthesis and photosynthesis or photosynthetic translocation, which is called as a source and sink relationship. So how it is going on in PGNP, to understand that, uh, um, I have intended this study, and in this study actually we are looking upon the basically photoperiodic pathway as well as the hormone biosynthesis pathway. In hormone, we all are aware that the gibberellic acid is very play important role in case of the um, cell elongation, that play important role for the tallness or dwarfness. But side by side, it is also important to manage the plant canopy. So there are so many other genes are also playing their role. So to understand the gene network, actually means I tried through NGS platform, I mean through this Galaxy, but unfortunately it is not having the plant genome, so I have restricted that study. But I did some analysis of that particular, or I will do this sequencing data, means from the differentially expressed data between the tall and open plant type versus to compact and dwarf plant type. And so what are the probable candidate genes? They are playing their roles, and if there are, means I come across some differentially expressed gene, I'll definitely go with the safe, with one example of GA20 oxidate. I have already carried out one exercise, and through string I have identified one candidate, that is GA20 oxidase. There are so many other GAs, but 20 oxidase is very important, and it shows the multiple order of interaction with the other molecule, and these are the probable candidates. And means through ranking, I've also carried out some interaction, and there are probable relation with the some entcorin and other related genes, and that I would use in future to understand the network. And ultimately this the network of this differentially expressed gene, as well as the metabolite, which includes protein also, and their influence on the pathway of GA biosynthesis. and how it manipulate the photoperiodic pathway also. So that will give you the clue about the manipulation of the plant type, and ultimately it will lead to the means we can manage the plant type, and we can make out the tailor-made plant type out of it, which is known as adiotypes. So thank you.
Thank you so much, sir. So
I
I
do have
see
one.
everyone
I do have
is
one.
very. Yeah, ma'am. Please.
Mr. Vipul, have you seen the BGP plant?
Actually, I work on PGNP only, means for I think last 5 years.
Okay. Uh, have you raised it in different locations and different months?
Yes. Because we are having the one main pulses research station over here, and I'm associated with that particular research projects, and in which we are having fortunately one compact dwarf and with the maximum secondary branch plant type.
Are you in which institute?
I am at Navsari Agriculture University.
Okay.
Uh, it is a
I'm
acid.
sorry. Have you means do you have any relationship research collaborations with IRA?
Yeah, we are having the AICRP Volunteering Center, in which we are conducting the many trials. Means one of my means many of my colleagues who are working in the plant breeding and genetics, they are handling the trial of off-season nursery of our IARI is advanced at Navsari only.
Yeah. We have shared some material with them
Yes.
through my IRA partners.
Yes.
Uh, but okay, I don't know the status of it. They are evaluating it for the past 6 years. No idea. But
Yes.
the thing is,
I think Dr.
when
Rajesh sir is there.
Sorry to interrupt for the conversation. I extremely sorry. With consideration of the time, ma'am, you can discuss.
yes, we will
So we
discuss
are
it.
running. Yeah, ma'am. Thank you for understanding. So we can proceed with the next presentation. Who is presenting next? Is it Ziaul? Uh, is is
I
your
can go. I can
network?
go for it. If
Ma'am.
Ziol is
Sure,
not
sure.
presenting, I can go. Yeah.
Yes. Please.
Yeah. So mine are I think from 6 to 10 were my slides.
Okay,
Can
ma'am.
you?
So, but we are
Yeah.
restricting 3 minutes for each presentation,
Yeah.
and 1 minute.
I completely
Yeah.
understand. I completely
Sure,
understand.
sure,
Yeah.
sure, sure,
Actually, I
sure.
couldn't
Thank you so
perform
much.
the test very well because I was in the middle of a meeting, and I just did it just for the sake of doing it. I am. I don't know. I don't have.
That's fine. That's
I
fine.
couldn't.
That's completely understandable,
Yeah.
ma'am. It's okay. Yeah. Thank you so
Yeah.
much, ma'am. You
Still,
can
I
please
am.
continue.
Yeah. Please
I
show
can
my slides.
understand.
I'll do it.
Okay.
Thank you. So I am working on plants, and my plant, which is guar, it is a legume plant. But since its genome sequence is not available, so I am not able to do much into it. So now in this workshop, I have tried some other plants whose genome is available. So basically, my topic is stress biology, how plants are tackling basically abiotic stress like drought and salinity, because even my crop was quite drought resistant, guar. So I did the whole genome analysis of a rice variety, disease resistant rice variety against a bacterial disease. Uh, I'm not able to see my slides. So I did
Ma'am,
it till,
you can
I
share
can
your
go
screen.
without them also.
Okay, if you're
Acha,
confident,
I
it's
have to
fine.
do it
But
myself. I'll do it without my slides because I'm doing it on mobile. So
okay, Dheeraj
unnecessarily
is
it will waste
presenting here.
time.
Just a minute, ma'am. Dheeraj is presenting.
Oh,
Thank you, Dheeraj.
thank
Thank
you
you so
very
much.
much. Thank you, Dheeraj. Thank you very much. So basically, because basically I'm looking to stress having a agriculture. Like, recently, even tomatoes they have become very costly and all this. So that is the importance of this work. Please go into the next slide. So here I have taken rice variety, which is the resistant. They have developed a resistant variety against this bacterial disease. Then I am planning to do meta-analysis for what is the role of annexin proteins in plant response to drought stress. And if a lot of data will be there, then I will be narrowing to millets, because millets, they are very nutritious, and 2023 has been also declared as an international year of millets, and they are very drought resistant also, stress resistant also. So that is why I have taken this, and the genome, the most important thing is genome of this foxtail millet is also available. So I did these three things, and I couldn't get into the results properly. I need to work on it further. It's in progress because I'm stuck in all the projects. I'm stuck into some places, and I'll be um, carrying out this work further. So I don't have much results to present, but I have done till um, SAM tools M pile up. There I am stuck. I will be doing it further for VAR scan and all. I will be doing, and my data collection for meta analysis also is on progress. I will be going forward to do this meta analysis. I think that's all I have to say.
Okay, thank you so much, ma'am. Thank you so much. Thank you, Dheeraj. So next, Ziaul, you can share your presentation. You can start presenting.
Uh, Hello everyone. Before presenting, I just want to say one thing that actually I prepared the slide PPT on my Microsoft Office, not on the this Google PPT. So when I'm trying to paste it, it pasted like an image on this Google slide. So if you permit, can I present my original PPT?
Ziyal, we made fervent requests. This is not accepted, please. We told you, we have been requesting you to, what is the idea of, having a collaborative work? I really don't understand. To save time, isn't it? We have, people here, who are spending a lot of time, volunteering, for recording.
So
What
I
is
have
the
already
idea of
pasted.
having, Google Slides online? You have to work in real time, and it would have been, we sincerely told you, to that, we'll be closing the link, by 6 p.m.
So listen to me. So I already pasted the means prepared Google slide as well, but the image quality is not good. That's why I'm saying I can
I'm
present.
driving. I'm helping. I'm attending this. There are Indrani who have stopped this. There is Malvika who is missing her dinner time and she is recording. What is this fun about? I really don't understand. When do we learn discipline?
Uh, Sir, please, you drive carefully, sir. Ziaul, you can just finish off your presentation quickly, please, without wasting much time. Ziaul?
Yeah, I think Raj sir is not understand what I'm saying. That's why.
We understand, but please try to keep his point of view. Without much wasting them, you can present whatever you have.
Yeah, please
Please.
go ahead. Please go ahead. Yeah, please go ahead. Yeah.
Yeah, please. The entire screen is visible. You can please go ahead.
Okay, so shall I start?
Yeah, please, please, please.
Okay. Good evening, everyone. So uh, I'm going to present my presentation on the topic essential bioinformatics domain, a basic to hands-on overview, which is the my learning objective behind attending this workshop as a part of the mentor-mentee relationship. So this is the general background about the study that I'm going to present. Actually, uh, I'm not going to present any means complete story or a complete what should I say, the outcome of the study. I basically learned something which I'm going to present before you. So this is the details of that. So as I told, the learning objective behind this workshop is to have the overview of major bioinformatics domains before the start of the mentor-mentee cohort, which is the prerequisite set by the my mentor, Raj sir, for to attend, to must have attend this workshop for that. So the using the application of the workshop learning, that the tools that we learn in this workshop, like system biology tools, machine learning, whole genome sequencing analysis, meta review, data science, and others. So we have to means I have to use this in my ongoing research project. So basically, this high throughput technologies in molecular biology leads to enormous data, which generates the need of this integrated system bioinformatics approach. So I propose to work on a fungal virulent factors responsible for the pathogenicity in humans and the mechanism of antimicrobial resistance. Uh, so we use the tools like PPI network using string, gene card, gene mania, and all. So for the interaction analysis, which helps in the predicting protein function uh, and drug ability and of the protein of interest, and the molecular docking tools helps in predicting the best inhibitor for the drug designing. So, and NGS techniques used to get the comprehensive data of the organism genetic variation, which we can use in my case the to get into the mechanism of the antimicrobial resistance in case of the fungus Candida albicans. So these are the methods which I incorporated. I basically worked on the these four
Sorry,
techniques,
Zuri,
the
to interrupt.
domains. Yeah, yeah. Yes.
You're running out of time actually.
Okay,
You
I'm
can
just
just.
finishing within 2 minutes. So these are the methodologies in which system biology I use the. I first selected the protein of interest, that is the SAP4. It's a virulent factor. It's responsible for the degradation of the host tissue used by the Candida species. So it's basically a acidic hydrolase, which not only helps in damage of the host tissue but also helps in the drawing the nutrition from the host. So we followed this pipeline. We first checked it on the string, and the PPI studies followed. Next, I did the whole genome sequencing. This pipeline is not yet completed, so it's just on running. Then I did the molecular docking using the Autodoc Vina and visualized the. Actually, I docked the SAP4 with the gallic acid, and then visualized and analyzed the data using Chimera and Lickplot. Then I just go with the systemic review and meta-analysis, and I just formulate using these methodology. I formulated the title of the distribution pattern and antimicrobial resistance of Candida albicans in Asian population. So these are the results. This is the result of the system biology approach, that is a PPI network that I get. And as I told, WS analysis is whole genome sequencing analysis under process, so I'm not showing the result here. This is the result of molecular docking, and the title that I finalized for the systemic review is this is a systematic review on distribution of and antifungal resistant pattern of Candida species in the Asian population. The conclusion which I draw from this short approach is that biological sciences are not alone. An integrated system bioinformatics approach had multifaceted and ons to it. So SAP4 has a great potential to be targeted for drug designing, and phytochemical acts as an excellent source for developing antifungal. Systemic review and meta-analysis on distribution of antifungal resistant pattern in Asian population will definitely give significant clues for the future result. Thank you.
Thank you, Ziol. So we'll proceed with the next presentation. Deepika, are you ready?
Yes, ma'am.
Please go ahead. You can share your
You can share your
screen and start presenting.
screen and start presenting.
Ma'am, is it visible?
Yes, you can go ahead.
Okay, ma'am, just wait. Uh, Yeah, so my name is Deepika Goyal and I am a BSc graduate from Biotechnology Honors. So today's my topic is integrated bioinformatics study on INHA protein. That is a protein which is responsible for interacting with mycolic acids with Mycobacterium tuberculosis and causing tuberculosis disease. So the complete name is enoyl acyl carrier protein reductase, and this enzyme is involved in the synthesis of mycolic acid, as I already told, which are essential for the survival of Mycobacterium tuberculosis. Drugs that target the INHA have been shown to be effective against both drug-sensitive and drug-resistant tuberculosis strains. So this is the structure of the protein which I have taken from the PDB site. uh, the methods which I have been using are Cytoscape and Cytohubba, with the help of which I have constructed the node graph, due to which I can represent the different kind of nodes which were interacting and the different kind of genes which were interacting with each other. And the next is next generation sequencing, which I have done with the help of Galaxy, which is a server, and the SRR number is this, and it was a Homo sapien. It was a WES of Homo sapien male blood, and the fast QC analysis. The fast QC analysis was done also using Galaxy, and after that, I have done molecular docking against the curcumin, which is a protein which is extracted from the turmeric family, and the results were analyzed using Discovery Studio. So these are the results I have done. I have done in the hierarchical layout. So the most interactive gene with the INHA is the FABG2, which is most similar to INHA, and this is the fast QC result which I have got, and these are the molecular docking results which I have got. The ligand which I have used is the curcumin, which is basically responsible for reducing the effect of cytokinins and during the transcription, so that it increases the immunity level and boosts the cure from tuberculosis. So the conclusion is INHA have been validated as the primary molecular target of the frontline anti tubercular drug ionized. Uh, Studies demonstrated that INHA is also target for the second line anti-tubercular drug, and further studies and research is required to understand how this protein is important in the life cycle of Mycobacterium tuberculosis, and what are the other drugs which can be used to inhibit the function of INHA. Thank you.
Well done, Deepika. So you have gained the knowledge and that you have implemented in this work. Thank you, Deepika. You can stop sharing your screen.
Thank you, ma'am.
So next presentee is it Vijayanta Sarkar?
Yes.
Yeah,
I can.
please can you continue with the presentation, please?
Is it visible?
Yes, sir, it's visible.
Yeah, please go ahead.
Lots of work I have learned from this workshop, but some selective work I am presenting here, uh, with entitled different bioinformatic analysis of raw data related to metagenomic analysis and molecular docking. I am Vijayanta Sirkar, MPhil scholar, Department of Zoology, University of Gourbanga. And here the introduction of metagenomics. What is metagenomics? We all know that meta means The meta implies that the many organisms together, and metagenomics is usually used when we are studying microbial communities. And I have analyzed S RR230 48757 nanopore genomic reads of Lactobacillus plantarum using Galaxy platform, and another study on molecular docking. And I have selected two target proteins of methicillin resistant Staphylococcus aureus, alpha hemolysin, PDBID mentioned here, and bicomponent leukocidin, leucid subunit D. These two proteins are important because these cause the virulence of the selected bacteria. And one phytocomponent from the olive plant, oleoeuropin, was selected for the ligand for molecular docking study and downloaded from PubChem database. Methods we have used for I have used for metagenomic analysis, Galaxy platform, and molecular docking. I have used Autodoc Vina tools. Now the results and discussion. This is the par-based quality score, and then adapter content, then part sequence GC content, and the picture showing the cut adapt tools by which we can trim the adapter. And here is the target proteins of MRSA to protein, target protein here, and the ligand picture showing here, and the docking results is there, 3D interaction and 2D interaction. In 2D interaction picture, we can view that the van der Waals interaction, the conventional hydrogen bond, carbon hydrogen bond also there, and one lysine amino acid showing the unfavorable donor-donor bond. Here is the whole the result, alpha hemolysin and bicomponent liquid subunit D inhibited by this oleoeuropin subunit, the ligands, and binding affinity was very less, minus 8.9 kilocalorie per mole. And the conclusion is a molecular docking result showed selected ligand actively inhibit the target proteins, though more studies are needed to know about adequate profile of the ligands for future administration. I have followed several steps of metagenomic analysis using Galaxy server and molecular docking studies. This workshop was very helpful for me. Lots of steps are remaining to follow. If recorded session are provided day by day manner, that would be very good for the practice. And thanks to all. Thanks to all.
Thank you, sir. Thank you for your sweet words. Actually, yes, you can check all the recordings in Bioclues YouTube page. You'll get all the recorded sessions there.
Okay,
It's
thank you.
just a gentle message for all of the participants. Thank you. Thank you. So next, who else is left? Vipul sir, you're presented, right?
Yes, ma'am. Yes, ma'am.
Yeah, yeah. Thank you. Thank you. Just wanted to cross check. Okay. So if we're not left with anyone, Dheeraj, please.
Yeah, it's visible, right?
Yeah, yeah. Dheeraj, you can just start presenting.
Yeah.
Please
Um,
start.
Hello everyone, I'm Dheeraj and I'm a BTech graduate from Vignan University. I would like to share my project regarding this integrated system bioinformatics only. Like I've considered one gene mTOF, which is a mitochondrial transcription termination factor. So much of the studies are not there in this mTOF, so I've chosen this gene to study. Like we know, mitochondrial genes are very much important as well as the other genes also. So this particular mTOF is present in animals as well as plants as well. So coming to animals, there are only four. In particular to humans, there are only four mTOF genes that are sequenced till today, and there are only present four. So compared to humans, there are many mTOFs in plants. So we thought of comparative analysis between these two, like any function would be similar or what. We thought of sequencing those. And from this workshop, I've used string and cytoscape analysis and phylogenetic analysis from Mega. So the results are displayed like this, and this is my um, this is my phylogenetic analysis, and this is circular layout. All the 28 genes from the Solanum lycopersicum were added to this, and we checked out the interactions. And with the human mTORs also we have checked. I could not disclose that in here because of that privacy. And these are all the genes that have the string network I've exported into the cytoscape and checked it. These were the interactions, and these are the three mTORs that are found when search for mTOR in Homo sapiens in string. And the fourth mTOF gene was not explored. That is mTOF2. So we'll look for the possible ways. And coming to the conclusion, and there are many future perspectives in this, according to me. So that altering the mTOF itself is a transcription termination factor. So these help in transcription and initiation of the transcription termination as well as initiation. These genes helps in. So if there are any mutations or as any defects in the mitochondrial genes or in functionality, dysfunctionality, we can, if we can alter this, or if it is in initiation, we can change it. If we can change it to termination, like that might stop the mutation spreading. And like mTOP genes are present in mitochondria, right? Mitochondrial genes are as very much useful in studying evolutionary studies. So that I thought of studying further with the mTOF genes to gain the evolutionary studies in humans, which is related to this. Are there any mutations over the period of life from the starting? And if we could get the sequences also, that would be very good. And coming to this, only one SRA dataset is present in the NCBI also, that is in Arabidopsis. There's a much scope in doing this also. So I thought of considering this, and I'll end with this now. So I'd be very welcome to any suggestions for me to continue my research as well. Thank you.
Yeah, thank you, Dheeraj. All of you, I must say, like you made wonderful presentations. I understand there were some alterations or some last minute changes. Uh, so, considering all of this, but all of you has made a very wonderful work. I should request everyone to switch on their cameras. Um, Um, yes, so first of all, so concluding this entire 2 weeks continuous integrated system bioinformatics workshop, I take this immense pleasure and opportunity to thank each one of the participants to uh, expressing their dedication and making this as a successful event. And obviously, at last but not the least, since belonging as a joint secretary of Bioclues, I thank our advisors, Dr. Jayaraman Valadi sir, Sundarajan sir. Despite of his health issues, he has taken the pain to make it as a successful event. Uh, Followed by Prashant sir's talk on NGS and being a system biology expertise. Our outreach chairs, Anil sir, research chair, Pushpendra sir, our secretary, Manjari ma'am, each one of us has provided a lot of dedication, making their expertise, and have made a significant impact on on the learning process, so that each one of the participants can make it a successful event, which was very much uh, prominent from the way you have presented your work. That We feel so happy and glad to tell that you have all learned all these techniques, tools and techniques, but definitely this is not the end. If you want to continue your work and extend this as a like a proper research work, you should stay in touch with us. And what type of work that you are interested to do, please feel free to contact each of the mentors. And yes, it was a collaborative, enriching environment that made to travel through this workshop. And I feel like all of them have gained uh, skillful insights that have been immensely beneficial, and from the each of the expertise. So last but not the least, I should thank Malvika, Goldie, Dheeraj, for taking the pain and recording the sessions without considering their own hectic schedules. And obviously, I would like to thank our founder, Prashant sir, who has been the inspiration and the driving force behind all of the successful events of Bioclues. So I look forward to applying in taking this forward, like the knowledge that we have all gained can be made possible to successful future projects. So looking forward to all of you. If Prashant sir is there, we can have a photo. Prash sir, I don't think he's there with us. Just give me a minute. Malvika, you can also switch on your camera. Yeah, please do. Okay, Prash sir is driving still. Uh, yeah, we can also, we have our secretary, our wonderful secretary, she will be back at us with her wonderful stick, imaginary stick. So she will make work everything move in a fraction of seconds. Ma'am, can you switch on your camera, please? We would like to have a group photo.
I switched it on. I switched it on.
Okay, so everyone has switched on, so I can take a photo then. Smile, please. One more. Yeah. Thank you so much. Thank you, Manju ma'am, for joining the session and making it even more delightful. I thank Malvika. You can stop recording the session. Thanks once again. And I request all the participants, if you are interested to extend your research work in as for the successful projects or in making into research papers,